Alright guys, so this video is going to be our last lesson for the semester. We're going to finish everything up with gene regulation. And I introduced this a little bit in the beginning of our video when we were doing transcription for our diagrams. And so I'll be completely honest with you in that this is not my favorite lesson to teach. And it's partially because this is just one of those areas where it's not really fully understood by the experts. And so it's definitely not fully understood by me, your teacher. So I'll do the best I can. Remember that we don't have a test on this. You just have some questions on your semester exam. And your semester exam review should kind of point you in the right direction of the items that you need to know. So we're going to start with the concept of just DNA in general. And I mentioned this when we were talking about promoters and terminators, but not all of your DNA actually codes for protein. We know that DNA does hold the genetic blueprint, but about 97% doesn't actually code for proteins or RNA. It's just kind of what we call non-coding DNA. And for a long time, they just referred to it as junk, but I think they've kind of moved away from that term simply because there may be purpose for these non-coding regions. And so some of those purposes include things like the regulatory sequences, like the promoters and the terminators, the introns that are kind of interspersed throughout the genes. Not really sure of what their function is, but there's also some repeat DNA, and there are some areas in our genome where DNA is repeated, and they do serve a purpose there as well. So we do understand what some of these non-coding segments do, but there are some that it's still kind of unknown exactly why they are there. So some of this non-coding DNA comes in the form of repeats, and there are certain areas of your DNA where you'll find these repetitive sequences. A lot of times they're called satellite DNA if they're tandem, and what that means is they're together. And so you'll see the same repeats over and over and over again. You can also see that sometimes that these repeats actually can be disease-causing because of the way a particular segment gets repeated more than it should. And so when it says that they can be regular, mini, or micro, that just has to do with the size of the DNA sequence that is repeated. It's going to be more or less base pairs in length. Now this a lot of times is the area of our genome that we use for DNA fingerprinting. You hear about forensic study, and you're going to find that as humans, we're about 99% the same. Like, we are way more alike than we are different. So not even just within your family, because we're the same species, we have the same genes, and that we make the same proteins. We make human proteins, we have human organs, we have human enzymes, and so a lot of our coding DNA is pretty much the same. So a lot of the uniqueness that we have just comes in those little SNPs, or those little individual changes. So our DNA is way more alike than it is different. But one area where our DNA is unique is often in these repeat sections. And so if they're trying to do a DNA fingerprint, they're not going to compare the sections of the genome where we're alike. They're going to compare the sections of the genome where we are individually different. And so a lot of times these repeat sections are the pieces of DNA that they use to create a DNA fingerprint. They separate the DNA and look at the patterns. Now, sometimes these repeats can lead to genetic disorders. Like I mentioned, there's one called Fragile X, which is a girl. There's one called Huntington's disease, and it's where like a gene is repeated like 30 plus times more than it should, and so it leads to an overproduction of protein. And so sometimes it could have a negative effect. Now, one place where we'll often find these repeats are at telomeres and around the centromeres. And when we covered DNA replication, we talked about how that we need these protective end caps, these telomeres, because each time we copy our DNA, they get a little bit shorter, the chromosomes do, because of the whole primer and the DNA polymerase. And so by having these repeat sections in our telomeres, then it kind of protects us from deleting important genetic information. I'd imagine maybe the same is true for your centromeres, and that maybe those repeats there kind of protect us in terms of reading our genes. Now, sometimes the repeat DNA is kind of scattered throughout the genome. Not really quite sure exactly how that works, but you see the same sequences just kind of spread in different places. Now, one way that the interspersed repeats could occur is through these what they used to call jumping genes. We call them transposons. But there are pieces of DNA that are able to literally move within the genome. Now, sometimes they are physically cut with enzymes from one location and then they're reinserted into another location of the genome. Or sometimes an RNA copy is made and then it's turned back into DNA before it's inserted. But you can imagine that if you literally took a piece out and taped it somewhere else, then it's going to change the genome. If you're thinking about a paragraphs in a book, if you were to cut out a sentence and just stick it on another page, it may not make sense anymore. And so the same thing is true for our DNA. So depending on where these transposons end up, they could be disrupting a gene, or they could just land in some of that non-coding area. But there are ways that DNA can move within the genome, which can kind of shake things up a little bit. Now, when it comes to our cells, hopefully we know already, because we've talked about this a little bit, but we know that we all start the same way. We all start as a single cell when sperm and egg met. Now, genetically, we're different, but we all started as one cell, and then that one cell goes through rapid cell division, and it forms a ball of cells. And then at some point, our little embryos start to have parts. They start to have regions. They start to have organs. And so how does that happen? How do we go from this kind of blanket ball of cells that are all the same to these cells that have a very unique job, a unique structure, a unique function? And that's through the process of differentiation. Now, you have to understand that every cell in your body, every human body cell that makes up your body, okay, not your gametes, but your somatic cells, your body cells, every cell has the exact same set of 46 chromosomes. Because you started off as that one cell that went through cell division, all of your cells have the same DNA. Now, not all of your cells do the same thing, and so it has to do with which genes are expressed in each cell. And so what this lesson is about really is what determines which genes are expressed and which genes are not. How come some genes are turned on in some cells, and how come they're turned off in other cells? How come sometimes genes are turned on for longer periods in some cells versus others? And so that's essentially what makes a cell specialized, is it has to do with what proteins are made in that cell, what enzymes are made in that cell. And so there's a lot of different factors that regulate which genes are going to get copied. Now, normally it's going to be a protein thing, or it could also be due to factors that are outside of our cells in the environment. And so most of the time when we're talking about gene regulation, we're going to talk about it in terms of the transcription level, okay? Most of the time, the majority of your regulation is during the process of transcription. However, it could be regulated based on how we process that RNA, those edits that we make. It could be regulated at the translation stage, or it could be regulated after translation when we actually have our protein, and it could come down to how we modify our proteins. So some of our genes in our cells are always expressed. They're essentially kind of necessary just for human life, for to continue being a normal functioning human. And so these are called your constitutive genes. But there are some that are only expressed at certain times, maybe through development or at certain times in our life. You think about important stages like puberty or something like that, and some genes may only be expressed during that time, and we call those inducible genes. So in general, as I mentioned, most of your regulation is going to occur at the transcription level. So for the most part, if we transcribe a gene, which means we copy it into mRNA, then typically we are going to express that gene, which means we're going to make the protein, okay? So one of the big components of gene regulation are going to be those proteins that we talked about called transcription factors. So this is a group of regulatory proteins, and essentially they are going to determine whether or not a gene gets copied or transcribed, or whether it is not copied, okay? Now, typically they do that by binding to the DNA near the promoter. And so they essentially bind to those regulatory sections, and they will either encourage transcription or they will discourage transcription. Now, the ones that will promote transcription are known as activators. So these are proteins that are going to bind to the promoter And more than likely, they're going to allow RNA polymerase to bind, or they could be repressors. And a repressor are proteins that they actually prevent RNA polymerase from binding, and therefore they prevent transcription. So as we mentioned with our diagrams, the promoter sequence is where the RNA polymerase binds. It typically includes that TATA box, that TATA sequence. And essentially it's going to be just kind of like the precursor to the gene that tells RNA polymerase where to begin. It also tells the RNA polymerase which strand to copy. Because remember, in transcription, we're only copying one side of the DNA. And so essentially what has to happen is these transcription factors, they have to be attached near the promoter or at other regulatory sequences in order for RNA polymerase to bind and actually copy that DNA, okay? So the combination of the RNA polymerase and those transcription factors is called the transcription initiation complex. And so all of these proteins have to be in place in order for transcription to occur. A lot of times when we talked about cell communication, the result of cell communication is going to be the production of these proteins that may act as transcription factors, and therefore they regulate gene expression. So we can see up here we have our promoter sequence, we have our TATA box, here we have our transcription factors, these proteins that have to be in place. Once our transcription factors are in place, then RNA polymerase can bind and copy the gene. Now, the reason cells may do different things is because different genes are expressed, and so that could be because these transcription factors are only present in certain cells. So there's some that are only present in a specific cell type. Others may be present in all cells, but they have to be activated by other, chemical messengers or something like that. Now, enhancers are sequences. So these are actually DNA sequences that bind the activators. And then silencers are DNA sequences that bind the repressors. Now, a lot of times these are near the promoter, but they aren't always right there at the promoter region. They could actually be in another location of the genome, but through bending and moving, those sequences can come into contact. So essentially, the transcription factors are going to bind to these DNA sequences, and then they are either going to allow the gene to be copied or not. And so this differences in transcription is often referred to as epigenetics. So we know that our DNA determines our traits, but there are other factors that can play a role in the expression of our traits. And this is what epigenetics is all about. Just because you have a gene doesn't necessarily you express it or express it properly. And so these kind of changes that can occur, which can affect the expression of our genes, are known as epigenetic changes. And so a lot of times what they can do is they can alter the DNA in some way, not in the code, not in the sequence of the nitrogen bases, but in some other way. Or sometimes they actually alter the proteins that the DNA wraps around. And so depending on what kind of cell these changes are in, sometimes they could be passed on to daughter cells. And if they're in gametes, they could be passed on to offspring. So some of these epigenetic changes involve the state of the DNA, okay? So remember that most of the time DNA is in the chromatin form, and even in the chromatin form, DNA is kind of tightly compacted. It wraps around those proteins called histones to form the nucleosomes, and then that allows that 6 foot of DNA to fit in that little tiny nucleus. Now, because that DNA is tightly packed, even in the chromatin form, sometimes if it's too tightly packed, we can't actually get in there and copy it. Those proteins that are needed for replication, the DNA is essentially hidden. It can't be exposed to copy. And so one way that we can affect gene expression is by the state of DNA. And so if DNA is tightly packed, and one mechanism that can affect that is called DNA methylation. Now, one thing I want you to remember is that meth is bad, okay? So the reason I want you to remember that is that in this case, methylation, which is the addition of a methyl group, okay? But methylation is a negative effect on transcription. So we may not always want our genes to be copied, but in general, this is an inhibitory effect. So when our DNA gets methyl groups added to it, it actually makes the DNA more tightly packed, which essentially makes it unavailable. Okay, so it kind of turns it into this heterochromatin where it can't be transcribed. Okay, so DNA methylation is actually going to prevent transcription, which means it's going to prevent the gene expression. Now, another way that that DNA could be modified is by adding acetyl groups to those histone proteins. And if this happens, it actually makes the chromatin less tightly packed. And so what happens is it means those genes are more available to be transcribed. So methylation kind of inhibits transcription. Acetylation promotes transcription. Methylation is going to prevent the genes from being copied and expressed. Acetylation would help promote the transcription and expression of those genes. So notice we aren't making changes to the DNA sequence. We aren't mutating the bases or anything like that. But just by adding different chemicals onto that molecule, it can make it more or less available for transcription. So here's just a picture that shows you. Here would be when you had an acetyl group added. Notice it's not quite as tightly compacted, and therefore these genes could be copied. Whereas if we have methylation down here, it's kind of tightly packed and it can't be copied very easily. All right, so a lot of times these epigenetic changes, sometimes they could be permanent and they could permanently alter our gene expression. And as I mentioned earlier, if they're in our gametes, they can actually be passed down to offspring. So these epigenetic changes a lot of times are responsible for some differences. So for instance, identical twins, I mean, they have identical DNA. They started from the same egg and sperm that came together. So genetically, they should have the same 46 chromosomes, but yet there will be some differences between identical twins, both internally and externally. And a lot of times that could be due to these epigenetic changes. So a lot of times these epigenetic changes are due to the environment. So, our genes are great and our genes tell us who we are. But remember that environment also plays a role because environment can affect the expression of those genes. So things like extreme stress, exposure to chemicals, and this could be during development. So like if a woman is exposed to drugs or alcohol during pregnancy, or even as an adult, the things that you're exposed to can affect how your genes get copied and how your genes get expressed. All right, so far we've talked a lot about transcription and how transcription is going to be our main source of gene regulation. But as I mentioned earlier, there's other places where regulation could occur. And so one of those is in the RNA splicing. So remember, we copy our DNA, but within that DNA you have the introns and exons. And so a particular gene could be spliced in different ways, and that could lead to the production of different proteins. We could also have the regulation of mRNA degradation. So remember, the mRNA is made, it leaves the nucleus, it goes out into the cytoplasm. That mRNA could be read by multiple ribosomes, that mRNA could be read multiple times. And so depending on how long that mRNA is present, more or less of that protein could be made. And so one way that we can regulate like protein production is by determining how long that mRNA stays around before it's degraded. And so there's little microRNAs. They actually inhibit the translation of RNAs by causing them to become degraded. So these microRNAs could actually prevent translation and therefore prevent protein production. There could also be some translation controls. These microRNAs could inhibit translation or changes to those like 5' cap, the poly A tail. There could be proteins that repress translation. And so even if the gene is transcribed, if it's not translated, then that protein is not going to be made. And then there could even be further changes after the protein is made. So the protein itself could be edited or modified, which could lead to a change in expression. And so essentially it's this difference in gene expression that is going to lead to the specialization. So as I mentioned earlier, you start off as a single cell called a zygote. It goes through rapid cell division. Eventually those cells start to differentiate and become specialized, and then they morph or go through the process of morphogenesis, which is when they begin to organize into tissues and organs. And so early on in development, you don't have specialized cells. It's not until they begin to produce different proteins and express different genes that they become specialized and become our body parts that are essentially created to do a specific job. And so here's what ours would look like up here in the animal development. So you have rapid cell division or proliferation. It forms this hollow ball called a blastula. The blastula pinches in on itself. And then ultimately over time, depending on where these cells are located and which genes are getting turned on, these will ultimately become the different parts of the adult. So for a human, this is going to become different parts like our nervous system, our skin, our muscles, our bones, all of that kind of stuff. And so there's some other ways that differentiation can occur, and we're going to talk about this when we get to evolution as well, because sometimes evolution can occur as a result of changes in development. And so some other things that can affect gene expression have to do with just chemicals that are in the egg cell. Based on how they're distributed, that's going to have different effects on the surrounding cells. Part of it's due to cell communication. Remember that cells can secrete chemicals and communicate with each other, and that can maybe induce neighboring cells to make a change or something like that. There's also other chemicals called morphogens. They vary in concentration, and they can a lot of times act as transcription factors, which can either turn on genes or turn off genes. And then we also have some gene regulation genes. And so in animals, there's these genes known as Hox genes, and they're essentially kind of our master control genes. They kind of determine our basic body plan. And so, why is your head on one end and your feet on the other? Or why is your heart in your chest, and why are your intestines in your abdominal cavity? A lot of that just basic kind of body plan information is determined by Hox genes. And what Hox genes do is they turn on and regulate other genes, okay? And so a lot of times if you were to have mutations in Hox genes, it could lead to inappropriate body plan, like maybe you could have body parts in the wrong place. I've actually researched a little bit and tried to find what happens if you have mutations in human Hox genes. I would imagine a lot of those would not lead to a surviving offspring, but they have done some experiments like with fruit flies, and when they mutated the Hox genes, they ended up having a leg coming out of the head instead of an antenna. And so it just kind of goes to show you that the Hox genes kind of determine the location and place of body parts. And what they do is they turn on other genes and they regulate other gene expression. So for now, when you're studying gene regulation, it's kind of like when we did cell communication. These are just some of the things that are involved in gene regulation. We really aren't going into the specifics in any area. So the main things you need to know are just like that not all DNA is coding. What are some of the non-coding regions? You need to be familiar with transcription factors because that is a huge part of gene regulation. And you need to understand that, regulation is not always at the transcription level, but it can occur at translation, it can occur at the protein level. And so there's just a lot of components to gene regulation. And so if you just kind of understand this from just a basic level, that's really all you need for this class at this point in time. Like I said, we'll go into some of these things in more detail later, but we have officially completed our last set of notes for our fall semester. You're going to be doing an activity in your next class just to kind of help you practice with gene regulation. And then after that, you're going to be taking your quiz on your DNA replication and protein synthesis diagrams. So I'll probably be around in the next week or two. Don't forget you have surveys that you can complete for bonus. And I hope you guys have a wonderful day, and I will talk to you soon.